Hi, everybody. I'm Grace Walton from Hera Biolabs, and I'm happy to be here today. First, I'd like to thank the Cutting Edge Conversation staff from Scientist.com for organizing the Precision Oncology series. And today I'm going to tell you how our SRG OncoRat can advance your precision oncology work. Next slide, please. First, I'd like to introduce Hera Biolabs. We offer a suite of proprietary gene editing tools, including the piggyback transposon system and the Cas-CLOVER and Tal-CLOVER gene editing systems. And we provide these as reagents, and we also offer molecular biology services, including vector design, cell line engineering, and animal model generation. On the right-hand side of the slide, our in vivo work is anchored by our SRG OncoRat, or SRG rat. This is a highly immunocompromised rat that is a superior xenograft host. And we're currently using this rat to develop other models, including one with a humanized liver and a humanized immune system. We also offer a number of in vivo contract research services, including oncology studies in our SRG rat, other rats, and mice. And we provide animal model characterization and validation. So let me tell you a little bit more about the SRG rat. The SRG rat was developed on the Sprague Dolly background. It's a double knockout for IL-2 receptor gamma and RAG2. It's a SCID rat. However, it has enhanced immunodeficiency because it lacks B cells and circulating NK cells as well as T cells. In this rat, we get enhanced engraftment rates for PDXs and CDXs, as well as enhanced tumor growth. And then the nice thing about this rat compared to mice is that since it supports larger tumors in larger animals, we can perform serial tumor biopsies, serial blood draws. We can obtain more tissue at necropsy. And that allows us to combine pharmacokinetic, biomarker, tox, and drug efficacy studies. So here's an example of some nice data that we've obtained from the SRG OncoRat. The VCAP line is a prostate cancer line, and prostate cancers are notoriously difficult to grow in commercially available immunodeficient mice. The data on the upper left were obtained following subcutaneous inoculation of the same number of VCAP cells into the hind flank of SRG rats or NSG mice. As you can see, the VCAP growth is far more robust in the SRG rat than it is in the mice. And we often obtain roughly 10 times the tumor volume from SRG rats compared to NSG mice. And this is nice because we don't have to passage as often for PDXs or for cell lines. We can proceed to efficacy studies faster than we would in mice. And the SRG rats also offer the advantage for precision medicine that they accelerate the timeline from a patient biopsy to efficacy testing in animals. So back to the case of the VCAP tumors here, you can see in the lower left that the VCAP tumor in the SRG rat maintains androgen receptor expression and prostate-specific antigen expression after passaging. And then in the upper right, shows an example of how we're able to leverage serial blood draws. So these animals, as the tumors were growing, had blood draws weekly, and we measured PSA in the serum. And then we were able to see this nice tight correlation between serum PSA and tumor volumes as the tumors were growing. And then in the lower right, we show that the VCAP tumor is responsive to standard of care enzalutamide. Next slide, please. So now I'm going to pivot and show you a case study that's part of our ongoing effort to establish new PDX models. In this case, tumor tissue was taken from a patient biopsy and used to establish a PDX in the SRG rat, and we were able to provide actionable data back to the clinic to inform treatment decisions. So, working with our collaborators at the University of Kentucky Markey Cancer Center, we obtained a sample of non-small cell lung carcinoma biopsy from a middle-aged white male, and we used this to develop PDX 3010. We first inoculated the same amount of tissue, 8 cubic millimeters, into NSG mice or SRG rats. And you can see that the time to establishment and the tumor growth was much faster in the SRG rats. And then it's worth noting that from P0 onwards, next slide, please. From P0 onwards, we are able to obtain enough tissue to perform gene sequencing and histology. And in this case, we did histology on tumors taken from the rats at each passage, and we show that they remain positive for TTF1 and negative for P40, indicating that they are indeed the adenocarcinoma and not a squamous cell carcinoma. So after establishing tumor in the SRG rats, we then passaged back into NSG mice for efficacy studies. We proceeded to test standard of care treatment against PDX 3010 with cisplatin and docetaxel, and we only observed a moderate response. Then the bar chart on the right-hand side shows percent change in tumor volume in individual mice after starting cisplatin and docetaxel on day 0. Since we didn't see a sufficient response to cisplatin and docetaxel, we proceeded to perform genetic characterization of the tumors in an effort to identify efficacious treatment strategies. So our collaborators in Gautam Narlath's lab at the University of Michigan performed targeted exome sequencing and identified a mutation in the MET gene, which was a tyrosine to histidine substitution at amino acid 1248. And this was interesting because substitutions at this tyrosine have been reported in papillary renal carcinomas and are believed to be likely pathogenic. So because we thought that the MET mutation was pathogenic, we wanted to know whether MET inhibitors would inhibit PDX 3010 tumor growth. And Dr. Narla found a 2017 publication that showed that the 1248 tyrosine mutation in non-small cell lung cancer did in fact lead to resistance of type 1 MET inhibitors, such as crizotinib. But these same tumors were sensitive to the type 2 MET inhibitor, cabozantinib. So based on these data, Dr. Narla and we hypothesized that PDX 3010 would be sensitive to the type 2 inhibitor, cabozantinib. Back in the mice again, in the NSG mice, we proceeded to test the two drugs head-to-head. After dosing for 28 days, we saw no effect of crizotinib, but we saw nearly complete tumor regression with cabozantinib. And then, as with the previous cisplatin study, the histogram on the right-hand side shows percent change in tumor volume in individual mice. Okay, so then to verify the molecular mechanism of tumor regression following cabozantinib treatment, we performed Western blots for phospho and total AKT and phospho and total ERK on the left-hand side following either crizotinib or cabozantinib treatment. And as you can see across the top, the crizotinib failed to really completely suppress phospho-AKT and phospho-ERK, whereas across the bottom left, the cabozantinib completely obliterated the phospho signal. And then on the right-hand side, the IHC underscores the Western blot findings, and you can see that crizotinib does not suppress phospho-AKT and phospho-ERK, while cabozantinib makes them almost undetectable. So in conclusion, the SRG OncoRats are a fantastic host for establishing PDXs, and they enable fast tumor growth, and therefore they shorten the timeline to actually be able to provide data back to clinicians. We've had a lot of success with difficult cancers that are hard to grow in mice, such as prostate cancers. And in the case of PDX-3010, we were able to identify a pathogenic mutation and then communicate that back to the clinical oncologist at the Markey Cancer Center. And then we are continuing to work on how the SRG rat can be used as a patient avatar to further enable precision medicine. And so I'd like to thank our collaborators. Dr. Gautam Narla is the Chief of Genetic Medicine at the University of Michigan, and his group did a lot of the molecular biology work on PDX-3010. Mark Evers is the director of the University of Kentucky Markey Cancer Center, and he works with us to obtain patient biopsies. And then Fallon Noto is our scientific director here at Hera Biolabs, and she's in charge of R&D and in vivo service. Then I'd like to remind everybody that our products are available through scientist.com. You can obtain SRG OncoRats if you'd like to just purchase rats, or we can design studies for you that we carry out using the SRG OncoRats. We have PDX lines established from lung and ovarian cancers, or we can help you establish your own PDX. We do custom gene editing, including cell line engineering and animal model development. So thanks a lot, you guys, and we're open for questions soon, I think. Have a great day.